work that he's been doing on rice there at IRRI. Thanks.
Okay, thank you very much. So yeah, today I'll be talking really about the interface between genetic resources, plant breeding, and plant genomics, which is really what this conference is about. So it's really an exciting conference for me. And as Jeff said, I'll be discussing rice as the species. And for genomics, I'll be focusing, it's not just sequence data, but actually genotype data. So how do we use genotyping more efficiently to bring all of these discoveries down to breeding applications? So I'll be discussing the technologies, how to make more efficient, but how does everything tie together in the end? And so really kind of the classical molecular genetic approach we've been hearing is gene and QTL mapping, association mapping, and bringing it down to finding the genes controlling their traits of interest, finding the alleles, and then using that for breeding. And that's largely what I'll talk about, but there's some other approaches coming as well that kind of complement this approach. So in rice, we do have quite a diverse set of germplasm to start with, and there's quite a bit of work really across the community of identifying these genes of interest. And so how do we bring all those together? So I'll just start with, actually at IRRI, our plant breeding structure, and currently we have 7 breeding pipelines, and these include both irrigated, rain-fed, both hybrid and inbred rice. And we also have a number of trait development pipelines as well. So these are essentially groups working on specific traits that we've heard a lot about already, from the abiotic and biotic stress, grain quality, and then using largely that approach. So in rice, there actually are quite a lot of large effect genes that we can use. And so even the complex traits like drought, we found very nice large effect QTLs that can be used. And so that's really been the approach in the past. But how do we take that further in the future? So we're discussing now SNP genotyping. So this, as everyone knows, is sequence-based, but also getting it down to the haplotype level. So essentially closely linked SNP markers, that pattern of variation then describes specific alleles of interest for the breeding programs. And essentially that comes down to really the genotyping techniques that have made this more efficient recently. So I'm just showing one example, for instance, an Affinium SNP chip that gives a very efficient way to do SNP genotyping. But the reason really that we switched, I mean in the past we were using SSR markers and various approaches. Really it's almost unlimited SNP loci. These are biallelic, so it's very easy to handle with the informatics and the databases, being able to merge across platforms. But what it comes down to is really the low cost. So as the cost decreases, we can do a lot larger populations, we can do things faster and cheaper. And so that really opens up genotyping towards the forward breeding programs, which in the past haven't really integrated fully molecular marker assisted selection. So I'll be discussing really how do we incorporate that into our breeding programs. So just a little review for the situation in rice currently as far as what resources are available across the community. I'm just showing a few examples. Early on we had the IRRI SNP resequenced as 20 varieties. Susan McCooch at Cornell kind of led a rice SNP consortium where they resequenced about 125 genomes. And then more recently the CAES, BGI, and IRRI consortium to start sequencing the rice gene bank, which has 3,000 genomes. But what that comes down to for genotyping is we're interested to validate those SNP markers. Many of those could be sequence errors or various artifacts. So what we want really is to identify high quality markers that are actually useful and informative for breeding germplasm. And so a lot of that happened early on using fixed array formats. So for instance, say like Affymetrix 44K or 700K, and then Infinium 6K. So all of these developed at Cornell for the examples I'm using. And what that does is it gives us a deeper set of validation data. So we have our SNP discovery pools then that have been tested across larger sets of germplasm. So currently the 700K has been genotyped on 2,000 rice varieties and accessions, and essentially that's formed the basis of a phenotyping network that IRRI is leading now to do a genome-wide association study with that particular data set. Whereas for breeding, something like the 6K makes more sense because it's a lower cost. But what it comes down to really for the breeding programs to get the cost even further is we want more targeted like trait-based diagnostic SNP markers. Some of these being actually functional SNP markers or others that are essentially tracking those SNP haplotypes that are needed for the breeding programs. And all of this data then can be used for either trait-based or lower cost genome-wide assays, such as CASP assays, and the GCP had a very nice initiative to validate 2,000 CASP assays in rice, along with other subsets of SNP markers. And all of these then can be used really to accelerate and make more efficient both the upstream research and the breeding applications. So I'll just kind of briefly just mention an update on the 3,000 genomes in case some of you aren't really aware that this data is all entirely publicly available now. The raw data is available on GigaDB. So the GigaScience data publication then essentially described the data set and then made that raw data available also in the short read archive. And essentially it's 3,000 rice genomes and 14x coverage. So as I mentioned, it's really a collaboration with the Chinese Academy of Agricultural Sciences and BGI and IRRI, and a large team working on that. But essentially this gives us now really a great kind of valuable data set that we can start mining. And one way we're doing that is IRRI set up an International Rice Informatics Consortium. And so this is really an open consortium, so if anyone's interested, you can go to the website and get more information on how to join. We're really opening it up and really asking the community to assist in this effort. But the first tool that's really available at the moment is the SNP-seq database available on the IRRI website. So essentially, if you go to this website, you can go into the 3,000 genome data and tell it essentially which genome region of interest you have, and then what are the SNPs that are available. So this slide from Ken McNally just gives an example of, say you want to look into SD1, you can find 77 polymorphic SNPs across 2,235 varieties inside the SD1 gene itself. So this is something now that we really encourage the community to make use of, just because it's available now and quite useful. And so what I'll talk about for the rest of my talk really is how are we applying these technologies for breeding applications. And this is part of our Global Rice Science Partnership, or the GRISP program. And my work is really setting up the high-throughput SNP genotyping facilities at IRRI, and there's also one set up at CIAT as well. And then developing and validating trait-based SNP markers for breeding traits. And then finally, a SNP fingerprinting platform. And the way we set this up really is kind of like a core facility at IRRI, and is serving not just IRRI genetics and breeding groups, but also all of our partners. So anyone's welcome to send samples to IRRI, and the website has more information on how to do that. So recently, last year, we processed about 20,000 samples. Most of them are about a third on the Infinium 6K chip, providing about 32 million SNP data points. And so this has really formed kind of the basis of how we're improving essentially our facilities, but making it available to the rice community. And one way we tried to do that is to really optimize the workflow as well. So there's a lot of upstream effort to get the leaf tissue from the field, get it extracted, and do the genotyping. So I'll talk a little bit about how we're trying to improve the whole system. And a lot of this is being supported by the Transforming Rice Breeding Project, as far as getting it really to the point where we can scale up to the tens of thousands and hundreds of thousands of samples per year. And so one way is from Brooks Automation, the plant track system. So this is just a barcoded leaf puncher. So you scan and then punch leaves in the field so that it automatically remembers the identities of those samples. And then you go into the lab and you can use, for instance, a magnetic bead-based system. We're using LGC Octopure at the moment. And so it could do 8 plates in about an hour and a half. So trying to get essentially not only rapid extraction, but also very high quality. So you can go either into genotyping or sequencing like a GBS approach using the same DNA extraction. So we've kind of moved towards keeping high quality, but also very efficient extractions. But at the same time, trying to improve the plant growth facility as well. So really looking at kind of ways to grow high density seedlings in the greenhouse and integrating that with our genotyping workflow. So we can do for population development, there's still quite a bit of work done on marker-assisted backcrossing, major effect QTLs, and QTL pyramiding as well, trying to get really these into different elite backgrounds that'll make it more useful for the breeding program. So we're setting up really ways to improve and make that population development step more efficient as well. So now I'll talk a little bit about some of the platforms we're using, and essentially because there's so many different applications, there's different needs that often it does require kind of different platforms. These are just the ones we're using at the moment. There's quite a few more available on the market. We still do have some legacy SSRs and insertion deletion markers that we were using on a fragment analyzer. And then at the moment using Fluidigm, both 24 and 96 SNP assays. We now switched the Golden Gate to the Infinium 6K and then GBS as well. But really, I mean, it comes down to there's a lot of applications that can benefit from these approaches. And this includes really looking at a few really high value SNP markers, trait-based, really major genes that you want to profile in your breeding populations and track those SNP haplotypes, all the way down to quality control and seed purity, doing marker-assisted backcrossing, diversity analysis, up through fingerprinting, and then down eventually to GBV, so genomic estimated breeding value based selection, and really a lot of what people have already talked about in the meeting. So we're using these, and we're always exploring, you know, how do we get this to be more efficient. So at the moment, a lot of what we do is validating trait-based SNP markers. So in this case, we're looking at, there's a lot of publications that have information on major genes and SNPs at kind of key loci for the breeding programs, but many of those have not been validated in different germplasm sets or on different platforms. So we're now testing these markers and essentially developing breeder chips, which are kind of subsets of targeted markers that are trait specific. And so one example for rice, say you want to combine bacterial leaf blight tolerance resistance with sub1, the submergence tolerance. So you say you want to pyramid XA5 1321 with sub1. So we've developed gene-based markers for all of these gene-based SNP markers, and then flanking markers as well. So trying to essentially make it more efficient to say you want to transfer those into your into certain lines, or if you just want to track them in your forward breeding program as well. So I'll just give one example of how the sequence data is assisting in this effort. This is just some looking at sequence data across a number of lines near the XA13 locus, in this case using IGV, and then clearly identifying different SNP polymorphisms, but also a deletion event as well. So there's a lot of really having this wealth of sequence data has allowed us to really zoom into each locus and identify specific polymorphisms that we can then use for the breeding selection. So I think there's, like was mentioned earlier, it's kind of an overwhelming amount of data now that we have to sift through. And I think it's really the exciting part now is just having too much data at this point. But then at the same time, you can use that data to identify what are the most informative SNP markers for each locus of interest. And I think that's really where the breeding programs will start to benefit. And so at the moment, the platform we're using in this case is Fluidigm, but the result is the same no matter what platform. Is you'll get essentially these clusters. So in rice, it is an inbred crop, but we do have, in this case, heterozygotes. So this is, for instance, XA21. And it turns out that this single SNP is actually quite predictive of the wild allele at XA21 for resistance compared to the susceptible, which is across most cultivated germplasm. And so having, for instance, this gene-based SNP marker, then you can essentially use that marker to profile your breeding lines and tell which ones have the resistance at XA21 and which ones don't. And so really what we're trying to do now is increase the number of trait-based SNP markers so that we can have essentially very rapid profiling of the breeding materials for these major genes. At the same time, we're doing quite a bit more on SNP fingerprinting, and at the moment we're using the Infinium 6K developed at Cornell University by Susan McCooch and her group. There's actually 5,274 SNP loci that were validated from Illumina. We're getting about 4,500 that are really high quality across all of our germplasm groups. And we've run quite a few samples. And so it gives a very nice, really high quality data because the call rates are very high. So we don't have to worry about that missing data that, for instance, GBS provides. So a lot of people really like that at the moment. So they're using it both for high resolution mapping of populations, but also for fingerprinting diverse material. So in this case, it was really set up for the 5 subgroups of rice, but also a few of the wild species, such as Rufi Pogon and Nivara. So that's currently our main fingerprinting platform. But at the moment, we're also looking into GBS, as is everyone else. And so in this case, in the past, we had been sending samples out to Cornell for their service, as was mentioned before. So either 96 or 3D4-plex multiplex GBS on the High-seq. And we just ran recently a quick study just for bacterial leaf blight. So doing GWAS analysis with 285 diverse rice accessions. And so using the GBS data in rice, we get fewer SNPs than in maize, but 40,000 is certainly enough because we do have larger LD blocks in rice. And so we could really validate that we do have the known major genes, for instance, XA5, XA4, but then we have a lot of other potentially novel loci that we can use to follow up. So what we found really is GBS works really well, but there's quite a bit of informatics involved really for the QC and then filtering and getting it down to the point of, for instance, whether you use imputation or not. And you still have to deal with the missing data. So for the GWAS analysis, it works perfectly well. For other applications, we're still kind of struggling how to essentially merge or translate, say, like a 6K data set with a GBS data set as well, since most of the loci will be different. So there's quite a bit of more work involved with that. So really what we're doing is, at IRRI, there's still quite a bit of work on the marker-assisted backcrossing, like I mentioned. So if you take something like, in the past it was IR64, now we're using really the highest yielding modern varieties, the more recent releases. But essentially you have a whole host of large effect QTLs to choose from. So this might include sub-1 and pup1 for the phosphorus uptake, AG1 for anaerobic germination, salinity, drought, and heat, and essentially then combining them in different combinations. You wouldn't necessarily want all of them in one line, just because every time you do the marker-assisted backcrossing, you do have that chance for negative linkage drag. So even though we've tried to really make the introgressions as small as possible to reduce that chance, there's still some possibility of having some negative traits associated with those introgressions. So we tend to right now focus on the 2 or 3 QTL combinations. But that's still kind of looking at just taking an elite line and improving it. But the larger effort is actually in forward breeding. And so this is developing new varieties, which is really required if you want to increase the yield. So in this first approach, you can add some tolerance or some resistance, but it still has the same yield as before. So in this case, there's a large forward breeding program on all the 7 different pipelines I mentioned. And in these cases, you can start to use markers along each step of the way. So some of these, for instance, if this is, we're not using much DH, but we do have a rapid generation advance program at IRRI. So doing single seed descent through these early generations and then getting them out into the multi-location trials as fast as possible. But essentially there is a chance then to do some major gene selection early on in that rapid generation advance during the bulk selection process. But as was mentioned as well, there's quite a bit of work and value in genomic selection. So we're looking at using genome-wide prediction, but at the same time trying to combine the major gene selection into that model. So we don't want to really forget completely about all of the important genes that we know about in rice. And so trying to find a way to have a combination of the traditional genomic selection, but you include these major effect loci, for instance, as fixed effects in that model. And so that's something that we'd like to track. And really it comes down to having the informatics that can handle very large genomic selection populations, but also tracking those major genes at the same time. So I'll just mention some of the ways we're trying to approach that, and one is really on the breeding data management. So we do have a new system called Breeding for Rice, and this is our breeding information management system, again supported by the Transforming Rice Breeding Project. And in this case, it's a web-based information management system for the breeders. They've developed essentially kind of custom tool sets for the variety development pipelines, but also the trait development and cross-cutting teams as well. And so each of them, they have their own essentially customized system for study management. So you go out selecting the lines that you're using each year in the breeding program, but also a way to integrate the data, phenotype data collection. So trying to get that, for instance, tablet-based or kind of modernized phenotyping integrated into the system. So essentially everything is inputted real time so that you don't have, trying to avoid the process where the breeders have their own data and then they have to upload it later, which is always a struggle. So having essentially the database itself is where they upload it in the first place. And so that's really the key, is having not only the pedigree information, but all of the phenotype information essentially being used on a daily basis so that the database captures all of that real time. and so that, and then makes it available essentially to all the breeders who want to access it. So the hard part now is making sure it's interoperable with other systems, and this includes not only different phenotyping groups, so there's analytical services or grain quality lab, but also the genotype data. So we're now working at IRRI is trying to, how do we link up the genotype data so that it's kind of seamlessly works together with the breeding information management. And so that's, we've had quite a bit of effort recently on how to have the genotype SNP database work with essentially the breeding management database, and having it really as kind of an invisible, these things happen in the background. So the breeders don't have to worry about, how do I analyze that genotype data? It just is instantly available on a really almost a real-time basis. So this is kind of the work we're looking towards. Essentially then, so it's that data can be used for decision making for the breeders. And that's really, they've already released the first version of this, and they're continuing to improve the system. So I'll also mention that kind of related to this is a new initiative called GOBI. So this is the Genomic and Open Source Breeding Informatics Initiative, again led by Cornell, actually an Ithaca team, both Cornell, BTI, and USDA in Ithaca, and also 3 CG centers that will be part of this, including ICRISAT, CIMMYT, and IRRI, and really focusing on 5 major crops, so maize, wheat, sorghum, chickpea, and rice. And the key here is it's not really it's not an upstream discovery project, but it's really implementation for the breeding programs. So how do the breeders have that data at their fingertips, and how is it essentially all of that, all the calculations, everything's done, in the background, and so it just happens. And essentially that's kind of a vision that will take a number of years to reach, but it's great that we have such an excellent team working on it. I think Mark Sorrells is here, and then with the CG centers, essentially trying to bring what's the best technologies to bear on breeding data. And this includes, really the focus is on high density genomic data, which doesn't necessarily mean sequencing everything, but what it means is having imputation in place so that you have enough sequence data to impute to bring the cost down for your breeding populations. And one way this might work, this is a slide from Susan McCooch, who's leading the program, and essentially the idea is that we need prediction, selection, mating design, segment, tracking those haplotypes, and using imputation, and essentially having all of these functions linking to all the other databases that are of relevance. And it's not just a single database, but there's essentially everything needs to talk to each other. So between IBP, at IRRI we have our own, I mentioned Breeding for Rice, Katie Dart and DeepSeek and others, essentially having these, having the standards in place so that all the databases can talk to each other, and really leading it down to how do the breeders have that data, again, at their fingertips, like I mentioned. So this is really, I think, kind of the future, and this is probably the most exciting part of what we're working with. Essentially now the genotyping itself is routine, and I think the advances of the future will happen on the software development on the informatics side. So with that, I'll just acknowledge my team at IRRI. We do have working on the workflow optimization, marker validation, and GSL services, but also a lot of collaboration with Cornell University, with Susan McCooch. So with that, I'll take any questions. Thanks.
Thank you, Michael. Questions?
You mentioned about low-cost sampling as well as the data management. If a small seed company wants to invest in, can you give some idea about what would be the capital cost and what would be the running cost?
Right. So yeah, it's always hard to really give a number on the cost, but I mean the different platforms, it depends on the scale. So if it's relatively small scale, I mean just the handheld leaf punchers is enough. You say punching into a 96 format and going through. I would say on the infrastructure, a lot of what I showed, I mean, you could, if you had, say, like $500,000, I mean, you'd have a good start as far as some of the capital infrastructure. But really, once you get to the large scale, you're talking about, up to $1 million or more, several $1 million if you had a large company and really wanted to do large scale. A lot of these platforms, I mean, the more you spend up front for the capital, the cheaper is the running cost. So if you want the ultra low cost, both essentially genotyping, generally companies are paying more upfront for the equipment. Whereas if you, some of the cheaper systems up front, it turns out that the cost per data point will be more. So there's the balance between, do you want to spend more capital and have lower running costs, or if you want, some of the cheaper systems like Fluidigm is actually pretty reasonable, but then the cost is not as low as some of the other systems. So there's a balance there.
Great. Gary?
Could I pin you down a little
Yep.
bit more on
Sure.
costs? On your current structure, what are you charging? What would be full cost recovery on the 6K chip genotyping? And what is the cost of DNA extraction using your current line that you showed here?
Yep. So, and again, there's always, the internal, external costs and that sort of thing. So for cost recovery, but internal, I would say would be, it's this year now, it'll be $40 per sample for internal costs for 6K, but then actually external is about twice that.
And for DNA extraction?
Yeah, so the high quality extractions I showed, the consumable costs are less than $1, but the actual full cost recovery, we're up to like $2 to $3 per sample.
So this is way too high for forward breeding at those costs.
There is an issue with our full cost recovery methods that we're working with. So if you take just the consumable costs, it's cheap enough, but then there's a lot of other aspects that we need to work out. So that's something, something we have to deal with at IRRI, but essentially it depends how much you include in the calculation.
Yes.
This is Dhaliwal. After having demonstrated the utility of this, chip technology, when would you like to enter into the related genome or IRRI species? Like Novara, Rupipagan, Glabrima, etc. So for ABQ2L, that's where we have a lot of scope of improvement of, yield potential QTL, what we are looking for.
Yeah, so a lot of the work, once you go into wild species, you want to move away from the fixed arrays because most of them are designed for cultivated rice. So I think where a sequence-based approach is more relevant. Essentially because it doesn't matter if it's wild or not, if you sequence it, then you'll get the polymorphisms you need. But I think, for instance, Rod Wing in Arizona has done the most for the reference genomes for wild rice. But there's still a lot that could be done really to kind of integrate that useful diversity. So at IRRI, Dr. Jenna currently is working on the wild rice integrations, but there's still quite a ways to go with that.
Great. I think we have time probably for 2 more questions, if there's any questions. I'm going to look over here because my back's been to this group and I haven't seen if there's any hands up. So I just realized that. Is there anybody over here that has a question? Any other questions from the larger? There, we've got one.
I'm Deepak Santra from University of Nebraska, Proso millet breeder. My total probably project cost about $150,000 per year, where I have to pay 2 technicians, all these things. So basically, a plant breeder need to develop varieties, but not much funding for that. Now the number you just told, per data point, whatever cost, all these things, how many breeding program in developing countries can afford that kind of expense? And yet they have to develop varieties and meet the farmers' demand. So what should be the model to use this kind of sexy science, so-called?
Yeah, so that's a good question. So I think to the point that we'll see it actually affordable is when you reach, I think, below $5 a sample. Even down to, say, $2 to $3 per sample. Once it gets to that point where you have valuable, enough data to do what you want, but it's below that cost. But there's some technologies that can get there, but it would probably be more like a shared kind of central resource where you still be sending your samples to, like I mentioned, a lot of the very low cost technologies tend to be quite expensive for the capital. So if there's a kind of a shared facility, that would be the best model for that. But it is difficult if you try to implement a lab in every institute, because then you wouldn't have enough, like you mentioned, to bring the cost down. So that's with breeding, it's always comes down to the cost per sample, so that's something we all kind of struggle with.
Hey, do we have any last question? One more question. Okay, all right. Well, let's give Michael a brief applause and thank him.
Good afternoon. I am happy that most of you are still alert after 1 hour of lunch. And we have our next speaker, David Marshall, is going to speak on NGS variant discovery.